And so just to start off by saying, this session or workshop is really going to focus on how do you actually use our curation interfaces and some of the tools that we've built to help people do that. We're not going to go into great detail about how do you classify variants if you haven't already learned how to do that. We did provide some YouTube and other materials in advance, and we're happy to plan a different workshop at another time for that. But we hope even those of you who are relatively new from variant classification will get something out of sort of participating. We actively encourage you to participate when we get into the breakout rooms. During these short talks, I ask if people have questions that you put them in the chat. We'll probably have one of the ClinGen folks just try to answer in the chat, because we've really tried to devote most of the time to you working in teams with some of our great experienced curators who volunteered to participate today during the active part. So to just briefly go over the agenda for today, the first talk is going to be from Kevin Reilly, who is one of our software developers here at Baylor College of Medicine. He's going to talk about the ClinGen allele registry. The reason that's important is we understand from many of our colleagues in H3Africa, you're often trying to classify novel variants that may not yet be in ClinVar. So this provides you a way to find a unique identifier for your variants. They may already be in our allele registry. There's several billion such variants, but you can also register new ones. So Kevin's going to start off, then Matt Wright, who is one of the project directors at Stanford University for ClinGen, is going to introduce the variant curation interface itself, to sort of walk you through how it's put together, what it looks like, what are the different things that are brought in. And then Deb Ritter, who is a bioinformatician and faculty member here at Baylor, is going to give you an example of walking through a variant. How would you classify a variant in the BCI? At that point, I'm just going to give you some tips for how to work in teams in the breakout rooms. And then we're going to split you all up into 10 teams with our great curators who have volunteered to be leads in each room. And we have provided them some example variants that you can try on your own in the variant curation interface. And we'll talk more about that after the talks. So I think with that, I'm going to turn it over to Kevin. We do have people here monitoring the Zoom. We'll try to mute people if there's background noise. If someone wants to say something, please, if you can use the raise hand function, or just unmute yourself and let us know you had a question. But again, I think because the talks are relatively short, please put questions in the chat. And if we think, oh yeah, this is a question a lot of people have, we might stop and ask Kevin to answer it, or at the end. Otherwise, we'll just try to answer questions in the chat itself. And with that, I will turn it over to Kevin.
And I'm going to stop recording now. And Fareed, can you pick up the recording?
Yes, it's recording.
I'm stopping mine though. I wanted this to happen before Kevin.
Yeah, he is recording.
Great, thank you.
Great, thank you. Can you see my screen?
Yeah, but you need to go into slideshow.
Fantastic.
Yeah, that looks great.
Great, thank you. Thank you for the intro, Sharon, and thank you for Sharon and the rest of the organizing committee for inviting me to speak this morning. Like Sharon said, yeah, please put comments in the chat. I'm going to try to stay under my 20-minute window so I can address any comments or any particular variants or anything that anyone's interested in specific. So again, yes, welcome. Thank you for joining us. So I'm going to talk about the ClinGen allele registry today. So my name is Kevin Reilly. I'm a software engineering and programming manager at the Baylor College of Medicine, and I work on the ClinGen team. Okay, so first I'd like to give an overview of what I'd like to cover today. Starting off with the introduction to the ClinGen allele registry, I'd like to talk about the current status, the users, and the usage, following my existing functionality to support variant pathogenicity interpretation, but also generically just registering variants as well for people wanting being able to attain identifier for various usages. And then from next steps, where are we heading after this? So this is a citation for our publication from 2018, was in Human Mutation. And so the ClinGen allele registry is a naming service to provide globally unique canonical identifiers for variants. And so this is a foundation for aggregating additional supporting evidence information. And here's our website here. It's reg.clinicalgenome.org. And I have it on the footer of a lot of the subsequent slides, so you don't have to jot it down right now if you're interested in taking a look. Okay, so a little bit more about the allele registry. So I'm going to tell you a little bit more about this canonicalization step, and I'm going to show you an example on subsequent slides. But the canonicalization process across genome builds and transcripts, essentially it's aggregated variant data from many sources, condensed into a single identifier. And so this is useful for people looking at variants across different timeframes, 10 years ago, 5 years ago, last year. You have different variant transcript versions, as sequencing technology improves, and different transcripts are identified, and so it allows you to essentially condense down into a single identifier. And so what we're aiming to do here, per an FDA requirement, was getting stable and consistent variant identification and naming. So once these identifiers are minted, they never change, and so it's a permanent record. We also wanted identifiers on demand, so high bandwidth and low latency. That basically means how fast can we get this service and how stable can we make it. So we generally see between 1 and 100,000 queries per second, or registration as well. And so it really depends on the nature of your input, whether it's sorted and how far across the genome in transcripts it goes. But it's very performant thus far. It's free and open registration. I'm going to show you how to do that. If you would like to register a variant, you do not have to log in or do anything if you just want to query the allele registry. We abide by the FAIR principles: is findable, accessible, interoperable, and reusable. And we accept any valid HCBS or VCF variants. And an important note here is this does not require any external registration in other databases such as ClinVar. You can essentially mint a new identifier for a novel variant or something you've seen in a manuscript. So it's really agnostic of anything that exists in other databases. So you can use this for any type of purpose. I'd like to talk about the general usage first. So we generally see per month, we see around 6,500 users, a little more than 212,000 visits to the landing page, 181,000 UI uses, and 9.8 million API uses. And I'm going to talk about how we can leverage these APIs towards the end. I have an example for this, but you generally see that we have a lot more people using APIs than UIs. And you say, well, why is that? And so a lot of people use their own individual software, and they leverage our APIs to programmatically interoperate with the allele registry, whether it be bulk registration, bulk query, having a surface on your end that wants to query particular information we have for a little identifier, maybe within your website or different downstream software. And currently we have a little bit over 2.5 billion registered variants in the allele registry. And so we anticipate that number growing as we move forward. And you can see from our trends here, we're actually had a pretty large explosion of usage the last year in 2022. Specifically for our usage in Africa, and this is via Google Analytics. So you can see that we do have usage from a variety of, mostly from the northern and southern parts of Africa. So you can see our usage here, and this is basically, for the past year, this is the number of users. So you see a little under 800 users, and you can see the countries here that are represented. So, it's a diverse amount of countries, but we, of course, would like to increase the usage and utility service. Okay, here's an example of a canonical allele identifier here in the middle. And this is a messy slide, but I promise it'll make sense in a second. So you can see we have this identifier here in this red circle, red box in the middle, CA166157008. Okay, and what does that represent? And so this was minted from one of these sources here. It was likely a genomic or a transcript HCBS where someone says, I would like to register this variant. And we say, absolutely. And we give them this identifier. And through the course of looking at other databases and other import processes from these other sources, we see that this variant is represented in a lot of other external databases. And this is one of the important aspects of the allele registry, is saying, where else can you find information about this variant? And so you can see we have these 3 different genomic builds from 36, 37, and 38. We also have 62 transcripts that are represented by this. And you also see that we have the main select for the ensemble and NCBI transcripts here. And so that's something that you can also find in our structured data, the JSON. Beyond that, we have these links out to ClinVar for the variation ID, as well as the RCV IDs. We also have links out to dbSNP, myVariant.info, Nomad, PubMed identifications, as well as another, and I need to draw a line here, but for the evidence repository. And this is essentially the final published, variant assessment from the variant curation interface, which we'll be talking about a little bit in a couple of presentations. So this is what it looks like in the actual UI itself. And you can see the same things that we've highlighted in the previous section, as that we have your identifier at the top, we have link outs to the gene, we have our linked data, all the ones that I mentioned here in this top linked data section, and then we separate this out by the genomic and the transcript alleles. And so I removed all the other ones in between these two entities, but we have 62 transcript alleles, and I'll show you what that looks like as well. So I'm going to be jumping back and forth between the live version and not, but just this is the same variant here. So you can see that we have this top section here, and we have the gene, the link outs, the genomic, and here are all these transcripts that map to the same canonical identifier. I also want to make mention, we talked about APIs and interoperability. We also have this JSON LD here. And so you can get this all the same data in a structured version as well. So you can download this. You can also operate this interoperability via our APIs. So you can see that we have all of our external records here, the link outs to all these entities that I mentioned before. We have our genomic alleles, followed by all the transcript alleles. Okay, so creating a login is very simple. So if you go to the main splash page, you can either create, or if you forgot your login, you can select that. And so here's what it looks like when you're not logged in. If you scroll down on our main splash page at reg.clinicalgenome.org, you can create login, and it's a very simple process, and it's on demand. So as soon as you click that, you'll basically have a username and password that you can use to log in, and this works for the UI as well as the API. Our next step I want to talk about is the available queries that we have in the UI. And these are also available, accessible via API. And a note about that. So everything we do, we build based on APIs. And so then the UI just leverages our APIs. So if you see something that's in the UI, that's something that you can also automate on your end for bulk or a steady interoperability between the allele registry. And so let's take a look at a couple of these entities here. So once you have your drop down here, you can see that we have HGVS/descriptor. And so this is, if people are familiar with HGVS, it's basically any valid HGVS will work here. In descriptor, what that entails is essentially we also support copy number variants currently. I'm not going to be demoing that today unless someone would like to see an example, but you can see an example here. Essentially, it's a descriptor based on the build, your chromosome, oh, sorry, this one here. Your build, cytoband is optional, but we can use it. Your chromosome, your range can be precise or imprecise, and your dosage can be precise or imprecise. But following the slides here, we say, well, how do we actually register a new variant in the UI? Okay, so once you're logged in, you can actually, you can copy and paste this or something similar. And I'll show you some examples of entities that are not registered if you would like to do a demo on your own. So once we query this individual HGVS here, we'll see that it is not registered. And the way we know that is at the very top, we do not see a CA ID. And so I'm already logged in here, but what you can do is click Get Identifier, and it should be instantaneous. And so you see that we have this new ID. And so you can also see roughly how many canonical variants we have based on this incremental count. In this case, it's roughly 2.57 billion allele registry that's been registered.
And Kevin, I just want to interrupt to say the reason you need the identifier is if the variant's not in ClinVar, you can also curate it in the curation interface just with this identifier. So if you have a brand new variant and it's not in the allele registry, you can hit register. Once you have this number, you can then classify it in the BCI.
Absolutely. Thank you, Sharon. Yeah, that's a key point. And so all of our other downstream tools, which I won't be talking about today, will essentially start off with having an identifier to use. And the main rationale behind that is if people use an individual HGVS expression, and then you are then tasked with basically saying that this HGVS is similar to this HGVS, is similar to this genomic HGVS, and so it can be problematic on the database end, or people getting organized, or multiple groups talking about the same variant. So once you have your minted canonical identifier, all of these different variant representations, as well as shiftable representations, will go to the same identifier, which is essentially the same equivalent effect for a transcriptor and for the genome. And so you can see here, and so I have screenshots for these as well, just in case internet connectivity issues. But once you click Get Identifier, it is then minted. I also want to show you examples of if people want to see and test this out for themselves. I have a list here of other variants that have not been registered. And so the one that I showed was for 100 to 105 area of this transcript for deletion. But you can also see if you slowly increment the side, you can get a variant that has not yet been registered if you'd like to test it on your end. And then the reason I provide this type of direction is that, with 2.57 billion registered canonical alleles, we've seen, we've mostly saturated a lot of the protein coding genes. Just because something is registered in allele registry doesn't mean it's ever been observed in human. We have some databases that we've bulk registered that are predictive or in silico type of things that maybe have not been observed, but you would have an observation. You can mint a canonical allele identifier from that. So if anyone's interested in playing around with this, you can kind of use a deletion of various sizes to get something that's not registered.
Kevin, you got a really important question here, so I will interrupt you from the chat. Will other data, how quickly, I'll put it this way, will other databases know you've now registered that allele?
That's a very good question. And so currently, we have external databases that would that prefer to have other external identifiers such as the allele registry identifier. So we have some groups that automate their process. Every time they have a new observation on their end, they leverage the API to query our service to say, is this variant registered? If it is, give me the canonical allele identifier. If it's not, go ahead and register it via an API put action and then get the canonical allele identifier back. Other databases such as ClinVar are a little not necessarily synced up as well. Maybe periodically, they'll go and check. But what we're doing is we're working on some downstream processes that will allow people to subscribe to updates to the allele registry via a technology called, leveraging messaging queues. And so it is our hope that we can stay in sync with external databases, basically, on the fly and immediate, updates, as well as other databases can stay up to date with us. And so this is an important thing that we are focused on. We want to make sure that at least across ClinGen initially, and then across external databases, that we will completely synchronized. Okay. The next thing I'd like to talk about is a gene and variation-based query. And then I'm going to skip forward a little bit to kind of give this use case, and I'm going to go back to show what these examples are. And so we've also often seen like a biocurators that will come and identify a variant from a manuscript. And oftentimes they may or may not actually provide the accession for that particular gene. And so you have to do an interactive query to essentially narrow down the potential, identifiers that may be useful if they're already registered, or registering ones that may be correct. And so I'm going to walk through this process of if you cannot generate a fully qualified HGVS expression, we can still assist you. And so if we start on the homepage, you can see if you scroll down, we have this gene and variation based query. And so if we click on that, this is the example here from the slide. So it's TP53, and there's a C dot, and then it's a position of 743, and then the ref is a G, and the alt is an A. And so if we query this, we should get a response that basically says what is registered and what is not registered, and what are valid canonical alleles and what are invalid canonical alleles. So for this particular 743 position from a G to A, it says these are the valid. These are you can see it's for an identifier here. These are the ones that currently are registered that have this given canonical allele identifier. For this particular transcript, it says it's not valid because the reference dated it was outside the reference itself. So this reference transcript must stop before this position. And you'll see some other interesting things here, like you might find that a reference allele does not match at that position. So this is an important thing that the allele registry focuses on, is that if you provide the ref allele and it's incorrect for that transcript, we will not allow it to be a valid registered entity for two reasons. One, because it may be misleading, but two, it's also, you cannot provide canonicalization across things that you are not certain what that sequence is. And so you can see here, another point is that for most of these top ones, you find the same reference canonical allele. So all of these transcripts agree upon this position, and then the leftover alignment says that these things are in the same coordinate space. However, this one is slightly different. So that means that these things, it happens to be a G at this position, but the alignment between these things are different. So we constantly see these things, and the reasons usually like alternative splicing and just differences between these transcripts. And so what this allows people to do is basically be able to obtain a canonical allele identifier with the most information that they possibly have. I want to show a couple of other examples. So in this case, you can also do an indel query. So if it's not a substitution, a simple, you can basically click on switch to indels, and this is what you're looking at right now. So for this particular gene, we can say C dot, and it's 100 to 114 deletion. So if we do a query based on that, we say, it's valid, but it's not yet in the registry. So you can see for these given entities here, it's not yet registered. So this is another way that you can register variants, is by clicking the get identifier here once you're logged in. Once you click this, it should bring up your newly minted canonical allele identifier. So you can see which ones are already minted and which ones are not. And sometimes it's difficult to tell which it is, which particular transcript is being referred to. So we've had people that were able to find additional information in the manuscript, maybe the supplemental information saying, oh, this is a transcript or this is a gene version, or people actually have to reach back to the authors sometimes to say which particular transcript did you use. But sometimes they all agree. In this case, it's nice. They all have the same thing. And so in this case, people can be relatively confident that it's one of these transcripts, assuming that the paper was done in recent time. And so this is an example here. And so if there's time or people are interested, we can go through these different ones. But from this particular manuscript, we saw that there was mention of this TP53 743G to A. We already looked at that example. We also saw that this BRAF from 1799 T to A. But I also want to show you that you can also do this from the protein as well. So let's say they did not mention the coding version, but they did have the protein version. How would we go about doing that? And so in this case, you can see that you would actually select P dot in this case. The position was 600, and then it's val the glue, just double checking here. Yep. And so if we do that query, we should see something similar. And it depends on, for the query, for how long these queries take, it's directly correlated to the number of transcripts and, sometimes the number of overlapping genes in the region. And then we actually look at the results here. We can see that there are a couple already registered identifiers, and some of these are the same, and some of them are different. So you can see that different transcripts. So this that ends with 43 here, ends with 31 there. So you can see that some of these.
Well, Kevin, they're not only different because of the transcript. Just to show you the first two. The first one is a missense change, and the second one is deletion insertion that gives you the same protein change. So if you're searching on a P dot, it will often give you every variant that gives you that protein change. Then you have to know, oh, they were actually talking about a missense variant, and so you can pick the first one.
Exactly. Thank you, Sharon. Very, very, very important points. And yeah, the other point I wanted to make is exactly what Sharon started alluding to as well, is that you can get the same type of canonical identifiers in different ways. So you can imagine that if you have essentially like a deletion of 3 nucleotides, well, you can also think about doing an insertion deletion where you add a couple and delete some. So there's so many different ways to get the same canonical identifiers for shiftable entities, but the allele registry handles that normalization and process. And it actually allows you to use the same identifier and not have to worry about that yourself. And this is also really important for regions that have like tandem duplications and repetitive regions, strings of A's or T's and such.
So Kevin, I think because of time, we should probably finish up. I think you were pretty close to everything you wanted to
Absolutely. And then just let everyone know that we talked about everything is also built on APIs. And so instead of, directly, looking for this canonical allele in the UI, you can also do the same thing via API and just show you what that example looks like. So this is a curl get, very straightforward, not logged in, anything like that. You get all the same structured data that you're interested in. And so moving forward, our plans is to continue to augment the allele registry content with additional linked data from the linked data hub, namely. We want to embed as much content as we can on the allele registry variant splash page so that people can find out as much information as they can from a single source, and also same kind of thing built on APIs as well. We're planning on adding additional structural variant support beyond CNVs, some things about privacy features. People are interested in not having their data necessarily exposed in a variety of ways. But so anyways, we're heading in a direction where we want to continue to support the stability of the allele registry, but continue to support features as well. Additional questions, feel free to email me at really@bcm.edu. And there's a lot of people to thank, but we're out of time. So thank you very much.
So I hope people will try it again. It's just reg.clinicalgenome.org. And you'll see we give you one example of a variant that's not in ClinVar, but has an allele registry ID. Okay, so next up is Matt to talk about the variant curation interface that we're going to work on during the breakout. Looks great.
Oh, it would help if I turn my mute off first. So hello everyone, good morning to you all from the Pacific West Coast. It's a great pleasure that I'm going to present to you today the background on ClinGen's variant curation interface that we'll call VCI for short. So here's a diagram from the ClinGen website that shows the overall ClinGen mission, the primary goal of which is to improve patient care for everyone through genomic medicine, which it aims to achieve by disseminating expert curated knowledge. And as you can see, determining the pathogenicity of variants based on rigorous standards is one of ClinGen's core roles. This work is performed by variant curators such as yourselves, who are as part of their role of doing the variant curation, they need to collect unstructured data from various sources, for instance, from scientific papers, databases, etc. They need to be able to evaluate and structure these data by applying standardized processes, and then they need to a means to disseminate those structured, validated data in formats that are easily accessible, so that the expert curated knowledge can then feed back to support patient care. The VCI was developed by ClinGen's variant curation working group to support variant curators in their work. It was specifically designed to support the variant curation workflow you can see here on the left. I'll give a quick overview of that workflow now before I go into each of the steps in a lot more detail. So starting at the top, you can see the variant selection. The curator needs to unambiguously identify the variant they're working on. That leads into what Kevin was just talking about. The next step is evidence collection. The VCI needs to bring in, both automatically and by manual input, the available evidence about that variant. And this pool of evidence is then available for all biocurators within the VCI to see and use. Then curators make their own evaluation of each type of evidence by applying the ACMG rules. And then all of these evaluations are then combined to produce an overall classification selection to the variant. And for variant curation expert panels, this selection, this classification is then expertly reviewed and agreed before the final variant classification is ascertained and published to either ClinGen's evidence repository and submitted to ClinVar. So although the VCI was developed by ClinGen variant curation groups, it was always intended as a community curation tool. Indeed, the VCI is available for use by all variant curators. And in fact, of the 2,600 registered users, about 1/3 are not associated with ClinGen. And we would encourage all of you to register to use it. However, for today's workshop, we're not using the main production site or test site of the VCI. We've set up special training sites for each of your groups to use. So you've already had accounts and passwords created for you. But after this workshop, we would encourage you to go register on our main production and test sites. You will need to register on both sites separately. And to do so, you just need to go to the header, click on login, and the box on the right will appear. It will pop up, and then you just need to click on create account. To set up your account within the VCI, you just follow the instructions. While each of you need your own accounts to curate in the VCI, you can also request a team account within the VCI known as an affiliation. Members of an affiliation can all work together within the VCI, curating and classifying variants together. This team functionality is available to any variant curation groups that request it. You don't need to be part of ClinGen. Indeed, out of the 100 or so affiliations we currently have, 15 of them are from non-ClinGen groups, and 2 of them are based in Africa. So working as an affiliation, it will also allow you to use some of the ClinVar submission pipeline features that I think Deb's going to go into a bit more on the next talk as well, which is not open to individual users. That's only if you're logged in as an affiliation. If you'd like to set up an affiliation for your lab or curation group, then all we need from you is the name of your group, as you'd like it displayed in the VCI, and the details of the contact person for the group. Other details like who should be members can be added in later. And we've posted a link in the Google Sheet into the agenda for the workshop. So please go ahead and fill in your details and we can set that up for you. You can reach out to us later as well, of course. Once you're logged into the VCI, you'll land on this page you see here. At the top, you'll see a welcome message. If you're logged in as part of an affiliation, it will show in the parentheses after your name, such as hearing loss you see here. Otherwise, it will just show no affiliation. The main feature of this page is a table that shows all of your variant interpretations that you've been working on as a group or individually. And you can search this table for specific variants, and it also has filter functionality, so you can find the variant that you're looking for. You can also download the whole table if you want to work on it offline. At any time, if you want to continue creating a variant, you simply click on the variant title and it will take you directly to that record, so you can continue your curation. However, if you want to start work on a new variant, then you go to the header bar and click on new variant curation. So as I said earlier, the first step is to select the variant you want to curate, and you do this by adding in either a ClinVar variation ID or a ClinGen allele registry ID. Adding either of these will unambiguously identify the variant. For instance, all known names for that variant will be returned for you to check that you have the right one, that you put the right ID in. Additionally, we return a single standardized title from the allele registry, and that's the one that's shown on the right-hand side in blue at the top. And it's this same title is used for the variant across all the ClinGen resources. Using these IDs, the CID and ClinID, also allows us to cross-reference data so that we can bring in the correct evidence for display in the VCI. Once you've entered a variant to curate, you land on this variant record page. Under the big title for the variant, you can see a table with various links. On the left, there are links to variant ID sources. So it will take you to like ClinVar, the allele registry, or dbSNP. In the middle, there are links to genome browsers where you can go view the genomic context of the variant in those browsers. For instance, if you click on UCSC, it will take you to the UCSC genome browser, and it will actually show you a window 30 base pairs up or down from the position of the variant as well. So you can see the whole genomic context. And finally, on the right-hand table, you see details of your interpretation that you're working on. As your curation progresses in the VCI, this will get filled in with the disease, any classifications, and the overall status of your work on that variant. And at the bottom of that panel, there is a link to the audit trail, which will show you every edit that's been made to that variant in its entire history. Further below that, you see a banner with all the ACMG criteria on it. This banner currently has a white background, but these have useful color indicators when an ACMG criteria have been applied. And I'll refer back to this later when I talk about evidence evaluation. With respect to evidence collection, the main thing I wanted to draw your attention to is a set of 6 tabs at the bottom, which will I'll zoom out a little bit on there. I'll go over each of these tabs now in more detail individually. The first tab, the basic information tab, is the default tab that's shown in the VCI when you go into the record. So we'll take a look at that tab first. Under this tab is the background basic information collected together about that variant. This includes at the top a table of the interpretation that exists for that variant already within the VCI, if there is one. This has just one, but it's a way for you to quickly see if there are other groups or individuals working on that same variant at the same time as you. Below that, we import the overall ClinVar interpretation, which is the summary of the ClinVar submissions, and includes the review status and overall clinical significance based on those submissions. And then below that, we list all the individual submissions for that variant in ClinVar, and you can click out to those to get more information. Staying on the basic information tab, but scrolling further down, the next table contains links to external evidence resources. These are smart links, so they only appear if there is evidence available for that variant in the external resource. For instance, for this variant, there is data in LOVD and LitVar, but not in Civic. Below that are tables of the known RefSeq and ensemble transcripts that overlap the variant. For each one, we show which exon it overlaps, the resultant protein change, if any, and the overall molecular consequence of the variant. We also tag the main select transcript in both tables. The main select transcript is the transcript that's been agreed by RefSeq and Ensemble to be the preferred representative transcript for the gene. And it's this one actually that's used when we're in the literature when we're generating the variant titles. The next tab is the population tab. It's here you'll see the allele frequency data for the variant as ascertained from various population databases, including Nomad, Exact, Page, and 1000 genomes. If there are other population databases that any of you are interested that are publicly available, please let us know and we can work toward potentially adding those in for you as well. Population data is brought in automatically on the fly via API from each resource. Additionally, the VCI works out the highest minor allele frequency, also known as the pop max, from each of the individual subpopulations across all the resources that are available. So for instance, for this specific variant, the highest minor allele frequency is from the African population within Nomad. Under the next tab, which is the variant type tab, so called because within it, the sections for evaluating the data are split into sections according to the type of variant. Within this table, there are two types of data that are brought in automatically. As you can see here, the VCI returns predictors data automatically on the fly from various APIs. An example is Revel, which you can see on the top row. This is a meta predictor tool that was developed as part of the ClinGen project, and it's on a simple 0 to 1 scale, with the higher score showing a higher chance of the variant being pathogenic. Other predictors include SIFT, Polyplan, and CAD. And for each score return, we provide you with the range and what the impact threshold is. Not shown here, but in a table below this one, the VCI also returns conservation analyses, such as from PhyloP, FastComs, etc. Next up, we have the experimental tab. Most of the evidence is added to this tab manually from papers by curators. So curators added a PubMed ID for the paper where they found the relevant functional data. The VCI then returns the details for that paper, such as the title and authors, so the curator can make sure they're adding in the correct paper. They can then select the associated ACMG criterion and write an explanation about the evidence they found. The next tab we have is the case segregation tab. Evidence is again added in here manually by curators, but this time it's not restricted to just papers. Indeed, you can add in evidence from clinical labs, from research labs, clinics, databases, etc., by selecting the appropriate source from the pull down. Because some of these sources of data are not public, that's the reason why at the top of this tab you see a disclaimer message. And this acts as a reminder to curators that they should not be adding like protected health information about individuals into the VCI. As a general rule, all unpublished patient-specific data entered into VCI, which is not explicitly consented for public sharing, should be the minimum necessary to inform the clinical significance of those genetic variants. Once you've selected one of the evidence sources from the pull down, you'll be directed to this form, which allows you to collect the case segregation evidence in a structured way. So we support structuring evidence wherever possible throughout the curation interfaces, because structured data is more valuable as an output because it allows for better cross-referencing and searching of the final disseminated classifications. One way of structuring data is via ontologies. We use these wherever we can. So for instance, we support Mondo IDs for diseases and HPO terms for described phenotypes. All of the individual values have their own fields for collection, such as the number of proband de novo appearances, number of homozygous appearances, etc. And each of these values added into the table are then collected together. And this table is just for one evidence source. So once evidence has been added from multiple sources, then each of these values added from different sources are all collected together. And they're rendered into a single table at the top of the case segregation tab. And this table, like this summary table of the data, allows you to see the collection of all the case segregation scores and the comments about those scores all together in one place. So you can easily scroll through the data. And it also means that the same types of data from different sources can be summed together to get total scores across the totality of all the evidence. In this case, there are 6 individual papers that have been entered, each one with a single proband het occurrence, but these have been summed together to show there are 6 such occurrences that have been found in the papers overall. And then the final tab is the gene-centric tab, and it contains the general reference information about the gene associated with the variant. It includes constraint scores from EXAC, which will be soon updated with Nomad. Additionally, it contains links out to resources to find more information about the gene or its encoded protein. So in those tabs, I showed you the evidence that is collected, both automatically and manually, within the VCI about each variant. And the next step of the process is evidence evaluation. So the ACMG produced a set of 28 codes that support variant curators in evaluating evidence. Each ACMG criterion is a question to ask about the specific evidence. Some of them, if answered in the affirmative, make a variant to be more likely to be benign, those beginning with B, and the rest, if considered to be met, make the variant more likely to be pathogenic, those beginning with P. So how is this evidence evaluated using the ACMG rules? Well, I'm going to give population as an example. There are 3 ACMG codes that allow you to question the population data you see displayed in the VCI. This is like BA1, BS1, PM2. So for BA1, which is shown at the top here, it asks if the allele frequency is greater than 5% in any of the population data. And if it is, then it's saved as met, and the variant is considered to be benign. Conversely, the PM2 shown at the bottom, the question is whether the variant is absent altogether from the population data set. And if that's the case, then that criteria is considered to be met, and thus the variant is more likely to be pathogenic. Looking to the VCI, as I pointed out earlier, all of the ACMG codes are found in the header of each page in the VCI in a banner. And you'll note that currently all of these codes have a white background. That shows that none of these criteria have been evaluated yet. So each code is a link. If you click on it, it will take you directly to the location in the VCI where you can evaluate that evidence for that specific criterion. So for instance, for PM2, the population code we were just looking at, if you click on that PM2 code, it will take you to the population tab and to the specific section where you can evaluate that evidence. Within that same tab is the population evidence. And as you can see, this specific variant is not present in any of the population databases. So from this population evidence, and the evaluation of that evidence from the pull down selected, the curator can select that this criterion is met and write in an explanation. One further thing I should mention at this point is that each ClinGen VSEP has the ability to write their own specification documents that detail how the rules should be applied following, how should be applied or followed according to their specific clinical domain. Specification documents that are used can be associated with the variant by the VSEP within the VCI, and all of these specification documents are generated and stored in ClinGen CSpec registry. All of the available evidence types for each variant are evaluated against the ACMG criteria. Each of these individual evaluations for the ACMG criteria then combined together, following combining rules laid out by the ACMG. This then provides an auto-calculated classification for the variant. As the creator evaluates each new ACMG criterion, this auto-calculation happens as they continue this process and is recorded in the banner at the top of the record. You'll notice that the PM2, PP4, and PM3 codes that were evaluated as met in the above example are now filled in with a background color to show that these codes have been evaluated as met. Furthermore, BP4 and PP3 codes have a grey background. This is how codes are indicated when they've been evaluated as not met by a curator. So this bar provides a quick visual indicator of your progress as you mark like each of these codes is met or not met, and importantly shows what the current auto-calculated classification is based on your evaluations so far. These evaluations are also summarized in a table known as evaluation summary. As you can see, the top table shows the 3 criteria considered based on the evidence to be met, and the bottom table shows those codes that were evaluated as not met. It also shows explanations provided by the curators when they were making their evaluations. Evaluation summary tables represent a quick reference of the curations for a variant and are useful in the expert review process. Expert review happens outside the VCI. Each of the VSEPs has experts within the clinical domain for the variants they're tasked with. They analyze the evidence and the evaluations based on the evidence and determine whether or not the calculated pathogenicity is correct for that variant and the associated disease. After expert review, they may decide to modify the pathogenicity or pull down, and they could do that at the VCI so long as they provide their reason for the change. We also encourage the VSEPs to write a comprehensive evidence summary, provide them guidance on how to do that. So once a variant has undergone expert review, then it can be published. VSEPs can publish right away to ClinGen's evidence repository, the eRepo, so that their final approved classifications are made immediately available to the public. They can also generate a file of their variant classifications that can be submitted to ClinVar. And if you sign up for an affiliation, you too can make use of that, like ClinVar submission pipeline. However, only submissions from ClinGen VSEPs will automatically get the 3 stars in the FDA recognized tab. So finally, I'd like to show you where you can get further help on how to use the VCI. First off, I'd recommend you go to the header in either the production or the test sites and click on the help in the header. From the pull down, you'll see various resources. I would recommend that you check out the SOP for the VCI, but I would also especially recommend that you take a look at the help documents for the VCI. We recently got migrated and updated all of our help documentation. It's pretty comprehensive, and it's generally well worth a look if you want to see how to use the VCI. You can also reach out to us at any time if you have queries about how to use the VCI via our help desk at vci@clinicalgenome.org, or if you want further help after today on the variant curation process itself, then you can reach out to the variant curation working group at variantcuration@clinicalgenome.org. So that's all I have, Sharon. That was my last slide.
Great, and we're just about on time. And thank you so much about mentioning the help document. I forgot to do that. So today, you're gonna be working in teams on these special test ones that we created for the workshop. But the Stanford team has put a lot of work into these help documents, so they should really help you afterwards. Any burning questions before we turn over to Deb for the last talk, and then we'll move on to the teams. See, I lost my chat there for a second. I don't see anything. So Deb, Deb, your view looks okay, but there's slightly overlapping. Can you make?
Okay, let's see if I can figure that out.
Make the VCI side a little smaller. I wouldn't make the slides any smaller.
Okay, is that good?
That's good. And we're using a part of Zoom that lets you look at two
Screens.
at the
Yeah.
same time. Can a couple of people who are based in Africa let us know that your Zoom is showing slides on the left and the VCI on the right? Either thumbs up or just put a note in the chat, because I realize. Yeah.
Great.
Okay.
Thank
Awesome.
you.
All
Great. All right.
First time we're trying it this way. So.
Yeah. So I'm Deb Ritter, an instructor at Baylor College of Medicine and a program manager for ClinGen. So I wanted to just quickly here, we have some links, of course, that we'll share afterwards. I did want to mention that we really do have a great community curation training program as well. And so we can put the link in the chat here. But they do have an upcoming training as well in April. So if you're interested to get, another round of training or to kind of go through that with ClinGen, we have it through this community curation opportunity. So let's see, moving here. So we're going to just start small today to go through and just curate one variant. So a lot of what I'm going to be doing is kind of covering a little bit of what Matt talked about, but we're going to be trying to do it live at the same time. So we'll be doing it on a variant from von Hippel-Lindau disease, which is an autosomal dominant hereditary cancer predisposition syndrome. And you can see it affects multiple organs here. And so together we're going to curate this variant serine 65 tryptophan or S65W in the VCI. So this is a known variant and it's a known pathogenic variant. So why would you want to curate one of those? Well, one of the main reasons is so that you can learn how to curate and that you can, it's a good training variant. It has a lot of evidence codes behind it as well. So I have navigated here and I'm in the demo site right now. So I'm in my group, which is group 5. So all my friends in group 5, if you can, while you're doing this, if you could use it under the no affiliation, if you're trying to walk through it at the same time. And then I'm going to do it in the test site affiliation. Otherwise, if you're all in the test site affiliation with me, we'll be kind of writing over each other. That's what happens in an affiliation. You each can choose evidence codes affect the same variant that you're curating. But if you're in no affiliation, then you'll have a separate curation. So I'm switching to test site example affiliation here and then saving that. And we've done that. Okay. So the next part that we'll do then is that we just need to select to curate a new variant. And please, if you're following along or have questions, please, you're welcome to come off mute and just ask them as I'm going through. So to curate this new variant, we're going to go to the top here and click to curate a new variant. You can see on the slides, I have the ClinVar ID and also the allele registry ID. So you could use either one of those to get into, to start the curation. So for this one, the ClinVar ID is 43597, and then I click retrieve. And it'll show you here that this is the variant, and then you'll click view evidence. So this is a bit of a, a kind of squished view here, but it's pulling up all of this information. So here in this evidence view, you can't enter any information in the for the evidence codes right now. This just shows you the evidence, but it'll pull up, what's in ClinVar. It gives you a link to ClinVar as well. It shows the population data here, and in this case, there is none. It shows the variant types, etc. But you can see there's no evidence codes that you can enter into here. You have to go into this interpretation view. So if you click interpretation, it'll have this user agreement. And then now you're in the interpretation view, and you can see this on the bottom here is these tabs that, that Matt had shown. And we don't have any of them filled in yet because we haven't done anything on it yet. So let me catch up our slides. This shows in ClinVar where to get this variation ID. We talked about this in the allele registry. We've saved and viewed, and we're in the interpretation. So as we move through these codes, I just want to reflect that many of these have additional guidance from the sequence variant interpretation working group in ClinGen. So we're not trying to do a curation that is from an expert panel with all of the specifications on it. Today, we're just wanting to get the codes into the VCI and to go through a couple of those. But there is much more information from the SVI where they've provided further guidance for several of these evidence codes. So here we are, and one of the first things we want to do is we want to add the disease, and we want to add the inheritance as well. So for the disease, we use Mondo ontologies. It's just a way that we can specify which disease we're curating on. So in this case, the Mondo ID here for VHL is this number. So we'll need to collect this number. This just provides consistent terms and identifiers for the disease. Okay, and put that in and retrieve. Did anyone have a question? So they've retrieved it here, and we'll save it. So our disease has been added, and then we add inheritance. So in this case, it's autosomal dominant. And you can select adjectives as well. However, we won't do that today. We'll just leave it as autosomal dominant. All right, so we've added our disease and our inheritance. And then again, we're in this basic information tab. And let's start out then with entering an evidence code. So we can go to the population tab, and again, you can get to it through either by clicking up here if you know the evidence codes, and you can also get to it through clicking this tab here. So let's go here. And again, we have this population criteria, which is BA1, BS1, or PM2. And so in this case, if you look down, there is nothing in Nomad, which is a good thing because we want to get the PM2 supporting code. So for PM2 supporting, then we can click here, click down to PM2 supporting, and we also have an explanation. So ClinGen has some standardized text that we have suggested to use for the explanations and for the variant summaries. I do have a link for that at the end of the slides. Those are very helpful in order to keep variants organized and keep the language kind of the same in between the variants that you curate. So here it says this variant is absent, and we put Nomad and we put the version and save. So you can see once I've saved, then this PM2 is highlighted here. So it's shown that I have used that evidence code. All right, moving on, then we can look at computational predictors next. I'll just try to cover a couple of codes here as we go through. And again, you're welcome to stop me at any time and ask questions. But we'll also be doing this again as we break into the small groups into our rooms and step through it together. So here, these are the computational predictor codes. So one great thing about the VCI is that it pulls in the information from all of these computational predictors. So here's Revel, here's all of these other predictors. It provides some conservation data as well. So if you can see here, the Revel score is 0.9. And for our VCEP, for our variant curation expert panel, we use, I think, above 0.6 as our cutoff for Revel. And so here on the PP3 code, we can choose MET. And then again, supply an explanation. so that the computational predictor is above 0.6 and it's met, and then save. All right, and again, it shows this code has been saved. So let's do one more code. And again, I didn't plan that we would do a full curation here on this one. I just wanted to show, using the BCI and adding these in as an example.
Deb, do you want to scroll up and show what the auto calculation is?
Yes, we are. So right now we're still at uncertain significance. So we have a moderate code and a supporting code, and you can see here that there's two pathogenics here. Well, we have two supporting actually, because we downgraded this to a supporting code. So we have two supporting codes. So our auto calculated classification is still uncertain significance. So let's add our PS4 code, which is the population, case code. And in this case, let's see, we can add in, here we'll go to MET. And again, we have quite a bit of data from here that shows that in a database we use for our curation that there are 17 unrelated individuals that have VHL cancers and across multiple publications. So I'll just select one of these publications that we can add in as evidence with this curation as well. Do this, okay. So I'm going to save this as MET. We can check what our classification is now. Now that we've added this strong code with two supporting codes, our classification. So I'll add in here. I'm going to quickly also turn off my video. It says my internet is a little unstable. So
we lost you only for a few seconds. So
okay, great. There we go. Preview. So it pulls up the article here, and then you can submit it. Then it pulls up this tab here that Matt had shown where you can add in examples. And so in this case, I'll add in, I know from the background on this paper, there were two subjects in here, and you can put a comment as well with it. So you could say that there were, let's say,
I would just
two subjects.
Two probands that met VHL criteria.
Yeah, and then we'll submit it. And then this keeps this publication here associated with your evidence code. It also, notes the number of probands. It would add that up, and then your comments, any comments that you make on it as well. And again, you can add that type of data as well for, for laboratories. So you could add it through a clinical lab, or a research lab, or a database, and each of those will have an area on the form where you can specify, further information about where that evidence item came from. All right, and so let's see the variant type tab. I think after this, I can add in one further evidence code. So I'll add in the functional evidence code, and that'll give us a pathogenic interpretation. And then we'll go to showing how you can export it, or at least in part, to ClinVar output. So let's go to our PS3 functional evidence code. And again, there is more recommendations from the SVI on this, but at this point, we'll put in that it is MET, and that we do show that this variant significantly decreases the binding of VHL with preventing ubiquitination of HIF alpha. So I'll save it. And again, with this PMID, I could add that in here as this curated evidence, and it would then associate that with a comment. I could specify a specific table in the paper or image in the paper, for instance, and that would keep that along with that evidence code.
And we do recommend if you ever reference a PMID in a comment that you do add the paper down below. I know Deb for time is doing it backwards and doing the code first. Typically you add the paper in the PMID, then you say that paper meets the code, because then the VCI saves all the papers that are added. And it makes a list at the end of every PubMed ID you've used.
Great. So yes, so for time, this is all the codes I'm going to add in. There are several more codes. Again, this was a very well-established pathogenic variant. There's no question that it's pathogenic. So, but I won't add in all of the other codes now. We've already gotten to pathogenic so far. So at this point, then you can go and you can view summary here. And we can see that our auto-calculated classification is pathogenic. Again, it shows all of these codes and it shows the different evidence items that are with them, as well as the comments. It shows those not met and it shows those not evaluated. And then if you want to change the classification, you can. In this case, we don't want to. And we would also provide a summary here. And again, we have some structured language for this summary. In this case, I'll just put, this is a well-known. Okay, and that's just as a quick summary there. So we can save it. And this will show that it is saved, and this shows a provisional save. You can add any comments if you want to, if you need to come back to change anything about that classification. Then you can do preview provisional and submit it. So now we've submitted this this interpretation here, and then you can also approve it. So you can have an approved date as well. That, for instance, anyone else in the team has then previewed it. Yes, I see a question in the chat. Okay, I think we got it covered. Preview approval and submit the approval. So now in this case, it is shown that that you have created this approval, created this variant, and then you can either do a batch curation, which we won't go into right now, or you can generate individual data. Because this is a test site example, I think that it's not going to generate the data, but you would click this button, generate. Here we are. Yes, it is. Wonderful. So this, then you could copy paste this line into a ClinVar submission file and then submit to ClinVar. And again, you could do, if you do this with a batch curation, then it would have several lines at once that could then be submitted and copied to a file to submit to ClinVar with all of the associated information.
Right. So I think, Deb, in terms of time, we're not going to get totally into ClinVar submission.
Right.
I want to make a few key points, and then I want you to answer a couple of questions.
Sure.
And we're doing fine on time. We assume we would go a little bit into the hour. So the first thing to just say, nothing is automatically submitted to ClinVar. This is generating a document that you can then use. Sorry, I'll turn my camera. That you can then use when your lab is ready to submit to ClinVar. I know early on in the VCI, there were a lot of concern, like, am I going to hit the wrong button? And all of a sudden, this is going to be in ClinVar. What the VCI is designed, ClinVar is a completely separate database. It's managed by a different branch of the National Institutes of Health. What the VCI is designed to do, because we definitely recommend you submit to ClinVar, is to just make it as easy as possible. So if you classify the variant in the variant curation interface, it will give you the document in the format that ClinVar wants. It makes it very easy for ClinVar to take it up. But you should have no fear that you're going to hit some button and it is suddenly going to be in ClinVar. Similarly, you do know that the variant curation, the variants that are classified by the ClinGen expert panels also appear on our website. Those again go through several clicks by the coordinator of the expert panel before they are officially approved. So just want to sort of give people ease. And today, nothing is going to come out of the test. So do like whatever you want. But the great question that you got was related to that. Like, how can you avoid a false classification? So I think if you want me to take it up, we, in the end, when you submit to ClinVar, it has your name as the submitter. It is not submitted by ClinGen. And so part of it is you can do all this work in the VCI and you can correct things. It's at that final stage. So for the ClinGen expert panels, it's at that final stage when we're going to say, okay, this is the approved variant classification and we're going to publish it. That typically is only done by 1 or 2 people in the expert panel. And it's when everything's been sort of checked and approved. On the affiliations that, as Matt mentioned, you can join yourselves, again, nothing sort of goes to the outside world until you submit it to ClinVar. In terms of what other people can see the evidence. So Deb gave an example of entering a paper and saying there were 2 people with VHL on that paper. On the real VCI, that is visible to anyone else classifying that variant. They don't know that you said PS3 is met or not met. All they know is, oh, someone found a paper and they claim that that's what was in it. So I hope that sort of assuades the question about sort of false classifications. I would think there was also one hand up. Did someone want to ask a question?
So I just wanted to add to that as well about the, how do you ensure against false classification? I mean, I think one is the process of carefully reviewing through the evidence codes, having it in kind of an organized manner in the VCI. And then you can also have an audit trail. We didn't go over that in the VCI, but there you can tell who changed an evidence code and when. So that in and of itself, keeping that history and that provenance of the data and of the classification can help. If there are any issues, you can go back and see where it occurred.
Melissa, did you want to say something?
Yes, thanks, Sharon. I just wanted to ask one question. So the SVI specification document is very helpful, the one on the ClinGen website, in terms of guiding the adjustment of the evidence levels for the codes. But I just wanted to ask, what is your advice for those of us working on genes for which there currently aren't any published kind of specifications? I feel like we get to a certain point and then the curation just becomes so messy because we have to sort of end up defining ourselves internally how we're going to standardize things and what we have to do. Is your advice just to kind of document that and just proceed accordingly, or should we reach out for some guidance or just await gene-specific guidance?
So I have sort of 3 answers. So number one, it's great to hear that the specification documents are helpful. And I think we actually went through it quickly, maybe in the groups. I think I've asked the volunteers to show the criteria specification registry is now online. So if you want to know, do we have a gene? Do we have specifications? The most up-to-date specifications, like we discovered that the P10 variant I picked, P10 yesterday, posted new specification documents. And so now you guys are going to get to curate it with the new rules. So that's number one. For the ones where we don't, that's where the sequence variant interpretation committee has tried to give general guidance. So that was a slide Deb showed early on. The SVI website, and maybe Deb, if you could put that link in the chat, that says, okay, how should you apply PBS1 to any gene? How should you, someone already kindly put the article with ClinGen's recommendations about functional assays. And that's the article, but in fact, that information is on the website as well. So I would definitely sort of recommend, if you're struggling with a particular code, see if the SVI has guidance. We're continuing to add to that. Like we have a paper that's being worked on right now to give guidance about splicing. So those papers, or the SVI guidance, is designed to apply to any gene. We are continuing, of course, to add genes. So we hope to add the last thing, I guess, if you wanted advice, like let's say it's something really about a particular disease area. We currently have 14 clinical domains. And so, and those again are on the website. So for hereditary cancer, for example, so if you had a question about, can I use somatic cancer data? We have published on that for those of you who do hereditary cancer. But, you know, it's also true that you could sort of send that question and see if someone in the domain who routinely classifies those variants might be able to, might be able to do it. Okay, maybe we'll take one more. Is there one more question? Anyone else? But that was a great general question. Okay, so let me get everybody ready for the fun part, or what we hope will be the fun part. Okay, so sort of key points for the next step. So, okay, so I recommend you take a picture of this screen. I did this workshop in person in India, and I kept trying to write down stuff, and all the young people in the committees were like, well, just take a picture of it. So in case you forget the password, it's here. So as a reminder, this is a demo or test version. This little sandbox that Deb provided me is just to remind you, you're not going to harm anything today. You know, so if you want to say something's benign that we all know is, you know, pathogenic, that's fine. It's just to try different options. Nothing that's entered here is shared with the real interface. We will delete this later this spring because we really don't want you to get used to this space. We want you to work in the real one or the real demo one, but it will be available for about 6 weeks. As I think all of you know, you've been assigned into one of 10 teams. And those teams, most of you are grouped together by country. We couldn't do that for everyone, but you know, hopefully that will work out. As a reminder, to get into the test VCI, and you want to use this URL. If you're on team 1, you put the number 1 here. If you're on team 2, you put the number 2 here. That will take you to your team's own personal variant curation interface. That's important to do because you've only been registered for the one on your team. The login is your email address. Many of you nicely came in and practiced yesterday. The most important thing we discovered is that the email, exactly how your email was written on the form in terms of capital letters and anything that might have been auto changed, is how we registered you. So I believe Freed yesterday resent everyone an email saying, this is what your email looks like to us. Please try that login first. And the password is exactly the same for all 10 teams. H3 Africa, little a, 2023 exclamation point. So all 10 teams use the same password. So when you get in your groups, you'll see the login, you put your email, you put the same password, and to get there, you use this URL. As Deb mentioned, and each of your group leaders may do it a little differently, you can work independently or you can all work together so you can see what each other is saying, that it's met or not met. Mark is here. He is the brave person who created all of the interfaces. We're going to have a breakout room 11 for people that are really having technical problems or something unique about your computer or something. And Mark and Matt will also be in that room. And you can move yourself in and out of room. So if you have your own problem, you can't get on, you want to try to have them help, you can leave your room, go to room 11, get it fixed and come back. And Fareed and I will be in the main room. So if there's just any simple questions, we can probably answer. Okay, so just as a reminder, and you'll hear this from your leaders, you can use either the ClinVar ID or the allele registry ID. We provided you both for the test demos. You have to use a Mondo ID for the disease name. And again, you can search for that, but to make it easier today, we gave you most of them. So for example, the last group of variants are in P10, we gave you the Mondo ID, and you can decide about the mechanism as Deb showed. These are the variants we're curating. So just to give you an idea, we're going to do 3 breakouts. The first are 2 straightforward variants. One's a straightforward benign, one should be a straightforward pathogenic. Whether you get the likely path or not, you guys can decide. But the goal is just to get everybody logged in, able to select codes, able to look at like Nomad in the VCI. Again, as Matt said, we're bringing the data to you. You don't have to go out to Nomad to see it. It will be in the variant curation interface. The second breakout will be trickier because it involves splicing. So we'll come back together in like 20 minutes and everyone can just report back. How did it go? We'll have a couple of rooms each time. We'll ask one or two rooms. Let's just say we start with, I don't know, room 1 and room 8. By the end of the first breakout, identify someone who just let us know which codes did you pick and what did you decide. The second one is trickier. It's splicing. And also it's a nonsense mutation, but that has some reasons for you to think it might be benign. And again, then we'll come back. And then the third room is we just picked missense variants. As Deb tried to show, missense variants often have the most codes. So we provided your leaders the data. So we, the Rebel score you can see in the VCI. We told you what papers said about it. And again, you can try to walk through those. The goal is not to finish classifying a variant. It's just to get used to using the VCI, assigning evidence codes, and looking at what kind of data you need to enter versus what kind of data might already be brought to you. So with that, and this is just the data we provided your group leaders on the 2 missense variants. So any technical issues, we talked about that your email is sensitive to capital letters in your email. You all should have gotten an email yesterday from Fareed with what capital letters we had in your email address. This again, remember, change the number in the URL to your team number. Password's the same. You can curate alone. Any login problems, go to breakout room 11. Oh, and if you do need to leave your breakout room, try to stay in the meeting because then Fareed doesn't have to reassign you. So let's say you got to go take a phone call, you have to do something, you can leave your breakout room, but stay in the meeting, just turn off your camera and your sound. And probably the most important slide is the huge thank you. So the H3Africa team, of which there's only a few members of the team who helped us get this all together. The ClinGen folks, many of whom you've met, and some of whom Chang'an's one of the group leads, and Amani is our NIH lead that helped us organize this. And then big thanks to our VCI curators who will each be in one of your breakout rooms to help you. And with that, Fareed, I think we are ready to put people in rooms. So are you going to be able to do it automatically?
Yeah, I can do it right now. So
okay, so you guys will all be in rooms. If you don't get put in a room, the team numbers are the same as the breakout room numbers. So if you somehow are still here when he hits his magic button, just as long as you know your team number, you can get into the right room. And it is 9:20, so we hope to see you back at 9:40. I think the first one, we're giving you a little bit more time just to let everybody get on. And we will send a note to your breakout room when you have 5 minutes to go. Go for it.
okay.
And it will ask you if you want to join. You do have to hit like yes. Okay, so for those of you who are left in the big room, it looks like for most of you, Fareed tried to assign you.
Yes.
There should be somewhere on your screen where it says join breakout room. You may have to click around on the Zoom.
The first two, I don't know if, well, if you go under breakout rooms, Dr. Plan, the first two individuals, I reached out to them.
Yeah.
I couldn't get a name or email to
Well, so
assign them.
Joanella is one of us, Kevin's one of us, so Calda.
Yeah, that individual, I couldn't get the name from the list. It wasn't there, nor the email, and same for
The
Uh,
purple one, who
yeah.
also joined yesterday.
yes.
So everybody else that's on here is Kelly in a room? Kelly's here, but doesn't look as unassigned. That may not have joined. Reem, you should be in room 4. Can folks that are still in the main room either unmute or tell us in chat why you are still here? We're trying to get you into room. So Heba, what room is Heba?
I just assigned her to room 9. That's
Okay.
her room. Yeah, I think when people kept leaving the initial chat before, I had to reassign them. So hopefully now when they join, they can go back to their rooms.
Okay. So
Yeah,
So
just
if people
Kelly-
aren't attentive, then they're not joining the room. That's why they're still here, even
okay,
if after you've assigned them.
yeah, Kelly Robin, you should be in room 5. There should be somewhere on your screen to select join breakout room. Because we're not doing anything here. They may have also walked away. So
Yeah, I was about to say, they're not attentive to the screen then. This happens.
the one where it appears to be a symbol, that individual also joined yesterday, but we couldn't hear their audio. So I don't know, because there's not a name, we don't know who it is. I tried
I'm gonna
to ask yesterday for them to rename themselves.
yeah.
Because we don't know what it is, so we can't put them in a room.
I've also asked them to unmute it.
Yeah,
No response.
okay, I'm trying that.
Yeah, I actually direct messaged that individual. I haven't gotten a response.
Okay.
So
My bet is the 3 of them may have walked away for a minute. And okay, but that worked pretty well. Timing worked pretty well. So,
yes.
and I haven't seen anyone pop out to say they can't log in, so that's a good sign.
Right.
No, well, that in India, that was the biggest problem we had. Although in retrospect, this email, we didn't figure out the email capital thing, and I wonder if that in the end was what it was. We had a few people who just couldn't. But I didn't have anybody, although actually the people from Stanford were awake at 2 a.m. But we didn't realize that. So that might have been it.
Sharon, I just wanted to comment that I thought that went really well. All the presentations were clear, and I thought having the demo was also good prior to doing breakout. I'm gonna try to stay here so I can at least hear the first part of the report back, but if not, I may have to log out. We'll see.
No, we understand.
But yeah, just
And
wanted
if you
to.
want to go in a room, you can just join a breakout room.
Okay.
So
Maybe that, yeah, no, I think I'm gonna stay here and then wait. So, but very good. And you did have a good turnout. I don't know in the end how many people joined from all the ones that had been registered.
I want to
it was
say
actually a pretty
57.
high proportion.
Yeah.
I mean, there's a whole bunch of us here, so you have to
Mhm.
sort of subtract. But I think at some
Yeah.
point we were. I mean, Free, were you taking?
Yeah, I did have, I want to say I checked around 67.
Which out of 75 is.
Yeah, it's really good.
Yeah.
Great. I'm glad. All right, going on mute, but I will.
No problem. Yeah, I think for this one, I'll stay here. The next one, I may pop into a few of the rooms.
One of those individuals left, actually.
Sharon, did you want me to stay handy or just via Slack or anything?
I could be via
Okay.
Slack. It's up to you.
Okay. do
They're
that.
not going to get to the allele, the one that only has an allele registry ID until the second group.
Okay.
But feel free to just come back, or I mean, I can Slack you or whatever. If you have questions about that.
Yeah, and I've been following our Google Analytics. It looks like we only have about 5 people so far from Africa, at least in terms of IP address. So I'm going to monitor that and see if we find we get any spikes or anything. So,
Oh, during it. Okay, sure.
great, thanks, Aaron.
Dr. Plan, what time should I broadcast? Like maybe,
Well, I like that we started on even time. So we started at 9:20.
right?
So 15 minutes from, so 9:35. Should do it. 2 people rejoined. So Amar, are you having problems?
That's also the other individual that I could not find
Oh.
on the list.
Amar,
So
we were trying to figure out which team you should be on.
can you type in your full name and email maybe in the chat?
Or can you unmute?
Yeah.
And it looks like Rolando's Rolando, are you rejoining with a different audio?
Yes, because I'm gonna go into my car soon.
Oh, okay, sure.
Drive home.
No problem.
So
I was
then I'm-
going to say that, well, yeah, it probably doesn't work in Africa. The Baylor Zoom will normally let it call you, but it probably wouldn't call you. I just realized as I was saying it, like, yeah, no, that probably won't work. But
Yes, I'll try and stay on. But if I drive and there's network issues, then I might
okay,
get off.
Yeah.
Okay.
just make sure you don't connect audio twice, because it'll.
Yeah, I haven't connected the audio yet.
I feel bad for this one person, but they have to be able to unmute or put something in the chat.
Yeah, I'm not sure. I can't read. Maybe if that's their name written in Arabic, I
I
can't.
think it's a symbol.
Oh,
Not sure. We tried yesterday to find out.
yeah. And then,
And I've sent a request on mute, but it doesn't. I mean, sometimes people do join from 2 devices, and so it could
Right.
be the device they're using for the camera, but
so they might have already
What-
been assigned their room if they have like joined with another name.
Yeah.
So,
then their camera would still be here, but
right.
Kelly Robin is, oh, and Omar is connecting. Just trying to figure out if people are having trouble.
Hello, Amar.
Omar, are you having problems? Room 10. Who is the volunteer?
Recording in progress.
Sorry, I didn't realize I was still sharing. Oh, it's happening. Kelly, you're back here, but again, you were assigned to room 5. Since I can move them, I just hesitate to move somebody.
Are
Kelly,
you seeing, because then when I go into breakout rooms, I don't see Kelly's name under unassigned.
No, what I see
Oh.
under room 5 is
Okay.
Kelly Robin has not joined room 5, and it says I can move that person. But I don't want to move them without them unmuting. So Kelly Robin, if you can hear us, we could move you into your room. Hey, Amanda, how's it going?
And systematically. Okay. Well, we just got dropped out of our Zoom link, so out of our room. So we need to go back into our room.
Oh, what happened? Because you
okay.
didn't pop up here. Yeah, I guess they're back.
How do we get back to our room?
You
What
click.
room number are you?
Yeah, we need to go. We need to go back to room 2.
Okay, I'll assign to you.
Great, thanks. Great, thank you.
Okay. Yeah, sorry. All right, I'm just gonna. And I see Deb.
They might have lost connection or something, I would assume,
Okay.
or clicked a button.
I'm gonna join room 5 for a second. So as I sort of expected, they were still on the first one. So oh,
You're back in room, the main room.
no, I know. I was just letting you folks know. I'm gonna check another one and see if they're, before you send your warning, and see if they're also still on the first one.
Okay.
Because we could potentially give people a little bit more time for the first one. Let's see. Here, I'll bug William. Okay, they are also on the first one. Why don't you send a message that says, we'll give everyone 5 extra minutes. Could you move on to the second variant or to, I mean, realistically to 9:50? So Free, could you send that? We'll give everyone until 50 minutes after the hour because it's not,
Yeah.
you know, they're all different hours. Please move on to the second variant if you haven't.
Let me do that right now.
But Williams' room, they also were all able to log on and thought it was going fine.
Sent that over. Let's see.
So that showed up, but not the second part about please move on to the second variant. You want me to put it in the chat or you got it?
I just put it.
Okay.
Okay. It looks like the other person has left.
Kelly Robin is still here, but I think may have walked away.
Well,
Right.
I'm not seeing her. I guess she's walked away.
Yeah, she is listed. If you scroll down to her room,
Yeah, she's at room 5.
she's listed for room 5, but he or she, actually, I don't know what the name like Kelly Robin. But that person is also not in room 5. So my idea that maybe they were.
Okay.
Uh-oh, people are coming back on time.
Okay.
Shireen and Rama, we were trying to give people a little bit more time because it looked like it took a little bit. Did your room break up?
Yeah, we got to the break room.
Thank you.
We
And
can
Ryan,
we can
sorry.
hop up in the breakout room if you'd like.
Yeah, I mean, did you get through the second variant?
No, not entirely. We were mostly walking through the VCI in the context of those 2 variants, but
Yeah, we tried to send a note, but we probably didn't give you enough warning to give people another 10 minutes because several groups were still on the first variant.
okay, in that case, if
I think you can just rejoin.
yeah, if room 3 wants to hop back in, we can go back right now.
Sorry for the confusion, or you can take a 5-minute break. Okay, Fareed, I'm gonna just do a quick bio break. I'll be right back.
Sure. Shangon, is your group complete?
Yes.
Okay.
Yeah.
Did you get through the second variant?
Yes.
Okay.
Yeah, but the second one is really no rush.
Yeah, we
Yeah.
just wanted to give some of the other groups or
Yeah.
you know, we
Okay.
wanted to give them some time so they have at least another 10 minutes.
Okay. I have a question. So will we be, pull back automatically, or we should?
There should be a feature on your end where you guys can go back to your room.
Okay, so we won't go back automatically.
Yeah.
Okay, I see.
I
Okay, got
won't be
it.
putting you back in, but
Yeah. Okay, got it. Yeah, I was just worried we will, be put back
do you
automatically.
see on your end where you can enter your room again?
Like now? Yeah, I think I.
Under, I don't know what it looks like on your end.
Yeah, there is a join breakout room and
yeah, so you would go back to your room that you were assigned to essentially.
Okay, so okay.
Because we just had Ryan's room come back and they went back to do the second variant at their room. So just want to make sure you have access to do that.
Okay, let me. Yes. Okay, so I can rejoin the room, but I don't have other like ask people to go to the main room function.
But can you come back to the main room?
Yeah, I just went to room 9 and came back again.
Okay, so yeah, your entire team, you guys are able to do that on your own.
Okay. So yeah, I know the next section I don't need to finish that in a big rush, so we won't
Right.
report back automatically. Yeah.
Okay.
Okay. So it looks like rooms are coming back.
Yes.
Should we wrap it up?
Should I send another broadcast?
Yeah, why don't you send a broadcast saying many groups are done, let's wrap up in the next minute. And then you can dissolve them.
Farid?
Yes.
Hi, Shayan.
Right. One people told me that he or she couldn't see my screen, but other people are able to see my screen. So is there any, I remember room 11 is the like a technical issue room?
That's group 11.
Group
Or-
11.
But
Okay.
that they couldn't see your screen on Zoom, or they couldn't see the VCI.
I don't know actually, but other people can. So
So they couldn't see your screen share essentially.
yeah, like only one. We have 4 and 3 people can see my screen, but 1 couldn't. So okay.
That I think maybe more a Zoom thing. They may want to, if they exit all the way out of Zoom, we can reassign them to a room.
Yeah.
So they may need to quit all the way out of Zoom and come back. That sounds more like a Zoom thing.
Okay.
Right. Or they're on their connection or something.
Got it. Yeah. So tell them. Okay. Thank you.
And just so people know, anyone can actually join any room. The only thing we can't reassign is you in your VCI team. But
sorry, Farid, could you me to join the team 3, please?
Where's mine?
oh, Amay, welcome.
Yes.
You're in the break, so we will.
Okay.
We'll assign you. We're wrapping everybody up. So I think you can dissolve people. You can blame me for it.
Good one.
We gave people a few extra minutes, but then a lot of people have finished up. So I think we'll bring everybody back.
Okay.
Welcome, Amay. We had a great first hour, had a bunch of introductory talks, and then everybody just went into their rooms for the first round of trying it.
Okay.
So we're dissolving everyone back in to talk
Okay.
about their first experience.
I just wanted to make sure that I have not been assigned another login with the VCI because I have my ClinGen access to the VCI.
Yes, but this is a completely separate VCI. This is just
Okay.
for the workshop. So
Okay.
But were you on the, Fareed, did he get assigned a team?
Team 1.
Your team one.
Yes.
So when we
I-
break
I
up
have
again,
assigned you.
when we break up again, just go into the first room.
Okay.
Okay. So
I have my invitation already on the screen.
okay, awesome. So, oh, you can, why don't we have Mark? Look, I'm going to join 11 for 1 second. I know. I'm just going to close all rooms. I'm hitting the red button. So they get a minute. They just got the 1 minute warning. So
16
just so you know, Fareed, that's what happens.
Yeah,
When you hit the all powerful red button.
yeah, I
they-
wasn't sure if that would close the whole thing or not.
No,
but
no, that closes all the rooms and everybody comes back. So that's
Yeah.
what you do at the end of each session. But it gives them a minute, so they're not cut off in mid-sentence. It starts a clock. So they have 30 seconds. Okay. So all the rooms have just closed. Everyone's coming back. Thank you, everyone. I hope that went well. We may need to just remute people because they've been talking, previously. So, so hopefully that went well. For Matt and Mark, congratulations on the technical end. It seems like there were very few technical issues. We had a couple of web Zoom issues, but so if you guys want to, Matt in particular, you may want to just join a room next time, because I'm sure you'll probably be interested to see, and Mark, how people are, the questions people are asking.
Yeah.
So hopefully that went well in terms of just getting people familiar with logging in and selecting some codes. I think we asked room 1 and room 8 to report. So is there somebody from room 1 that just wants to comment on their experience with the first variant?
No.
Did you guys assign anyone? No, we'll just make sure for the next one, who was the team lead in room 1? Bree, can you tell? I don't have the list up.
Yeah, it is
It's Inyo, yeah.
Oh, hello.
Hi, Inian. So in terms of your group, were there any recurring questions that came up or any issues with the common variant?
Oh, no, not really.
So one issue we saw, I can say, is that the VCI, maybe this is due to the test instance, but we only could see genomes in there and not exomes. I can screen share if that's helpful.
Oh, that's because it's a UTR variant.
Oh, I gotcha.
So it's
Gotcha,
not-
gotcha.
So see, that's why I wanted people to ask. So that,
Great.
that variant is in the beta globin untranslated region. So it's not on an exome.
Oh, wonderful. Well, that answers it.
So sorry, Matt, that's correct, right? If it's not,
Yeah,
in
that's correct.
exome, it doesn't show up. Okay, so the also a couple of people asked about joining teams. You can join any breakout room. We can't like assign you on the VCI side right now because but we set it up so people could move. So if you especially for the people who are some of the H3 Africa leads who are here, you can bop between rooms. You don't have to stay in the room you're assigned in. But just so people know, because a couple of people asked about that. Okay. And then did any of the groups, team 8 get to the nonsense variant? Any comments about that?
We did, and we had an interesting question about the disease entity, and that Eman had, and maybe she can further expand on that. But she was wondering about, what about genes in which there are multiple disease entities associated? So what would be the Mondo ID that you would choose?
Right. So that's my favorite question. Heidi and I fight about this all the time. So that's why I think it's really important. You'll see that we sort of gave you the examples of selecting the Mondo ID early. Turns out the VCI is set up, you can get actually pretty far before you have to select the disease. I think it's really important that you early on say, what disease am I classifying this variant for? So for example, I'm in the cancer domain. There are a number of genes that have both a developmental disorder, SMARCB1, right, can give you Coffin-Siris syndrome, and they have specific variants for that. Or it can give you rhabdoid tumor predisposition syndrome, and those are loss of function variants. So I think it's really important early to pick the Mondo ID you are curating on. Now, the gene I gave you is easy. If you actually take CTRAP, and I don't know, Deb, maybe you could pull up Mondo, and you just put that gene name in, it's the only disease that pops up. So it's pretty clear that that gene is associated with OI type 7. If you put RET in, which I also tried the other day, it will give you multiple endocrine neoplasia 2A, multiple endocrine neoplasia 2B, and it'll also give you Hirschsprung's. And Hirschsprung's is a loss of function mechanism, and multiple endocrine neoplasia 2A and 2B are gain of function. So obviously you're going to pick totally different codes. You would never pick PBS1 for RET for MEN2. You would pick PBS1 for RET if you were doing Hirschsprung's. So I think it's really important so everyone's keeping track. And that was the question about misclassification. I think you need to know what disease you're curating on. Just to give you sort of Heidi's view, and Marina probably could say it better than me, which is that sometimes you just have variants and you just want to start looking up the variant. Is it common? Is it rare? Is it a nonsense variant? And so some people will do some early curating and assigning codes before they've decided on the disease entity. But the VCI is set up to do Mendelian disorder classification. So it assumes that there's a gene disease validity for that relationship, and that's what you're classifying. So I think given that most uses of the VCI, it makes sense to pick the disease early on. Okay, so if it's an unclassified or a new syndrome, that's the issue. This is, for those of you who attended our last workshop, our last 2 or 3 day workshop was all about what do you do if you have exome data. The VCI is really set up to say, I already know something about this gene and this disease. And that's why we gave you sort of beta globin and said, curate it for thalassemia. That's a good example, right? There are variants of beta globin that can give you sickle cell. There are beta globin variants that can give you methemoglobinemia. Those are all different diseases, and you have to sort of know which one you're classifying on. This variant's a good example, however, for Heidi's example, is it's really common. It's going to be benign for all of them. And so that would be the argument why sometimes you may want to just start out and say, hey, this is a really common variant. It's not going to be pathogenic for any of these rare disorders. But if it's a rare variant, you're going to have to decide in beta globin, is this loss of function and it's going to give me thalassemia? Or is this like the classic, sickle cell variant? And so but it's not, the VCI is not really designed for gene discovery. It's designed for classifying variants in genes that you have knowledge cause a Mendelian or monogenic disorder. Okay. So I think because of time, we want to make sure you folks get at least the second room and at least a taste of the third room. Now, the second room has splicing. Splicing is really complicated. The VCI links you to different splicing algorithms. And as you'll see in the examples we gave, it's not always clear cut. So one of the examples is a very well-known pathogenic variant based on good RNA data. And you can sort of see what the predictors give. The other one is, much less certain. So The third variant, and so again, it's not designed for you folks to go all the way through. It's really just meant like, hey, let's look at the splicing codes this time. I mean, you're still gonna have to enter the variant. You can select the disease if you want. I think, again, we gave you the Mondo ID, but I would just sort of move right into sort of the codes for the first two related to splicing and sort of see what those look like. The third one in the group is just an example where I will say we don't visualize it in the VCI that well. You may want to look at that variant in ClinVar or in Nomad because you'll see what a FAR 3' nonsense mutation looks like and what data you might have about nearby benign variants. So those are the next three, and it's 10 o'clock, so we'll try 20 minutes, or maybe should we do, we're probably okay to try at 20 minutes, we'll give you a warning, but then it'll be sort of a 3-minute warning and we'll wrap it up. Okay. And as I mentioned, Mark and Matt, I think you folks could just join rooms if you want. Doesn't seem like we're having any technical problems, thanks to all the hard work you did in advance.
Cool, sounds good.
And again, if you don't get put in a room, you can join a room, but Fareed is going to do his little magic and assign you. And just as a reminder, you have to accept. So somewhere on your screen, it will say you're being asked to join this room.
Yeah, people are slowly.
Yeah, they are. Just always want to remind people that you can force somebody in, but the default is that it asks you. And Fareed, you do have some kind of roll call because we're going to give out certificates.
I do. I have, it's on the Excel spreadsheet.
Awesome. Okay. Then we'll work with Roland on that. Okay, so for you folks still in the main room, I know you might have walked away for a moment, but it should be asking you to join your room. Faruz, you are in room 10. Melissa is just Melissa's an H3Africa person. Melissa, you can join or not, that's up to you. I mean, she helped organizing this. Safa, again, you're in room 4. I think, and then Kelly Robin, who I think probably, probably it's sort of a ghost connection that they had to leave or something and it didn't disconnect. And since the meeting's still going, it'll just stay connected.
I actually just saw a message that Kelly Robin sent to the help desk.
Oh, yes.
Trying to log in, it keeps saying the user does not exist.
Oh, okay.
So that was,
let's see where
that was from about 10 after 10. So
yeah, no, that's great. So
yeah.
where is, because that person is in the main room, but we unfortunately I think is not able to unmute. Where did Mark go? Did we lose Mark?
I was going to say, I'm not sure which room he went into.
No, yeah, I don't see him. He might have left.
Oh, found him, room 2.
Okay. I can go into room 2 and just tell him.
Okay.
I could also reach out to her via email with the directions. I mean, I sent the email out yesterday about, the case sensitive letters, and maybe some people have missed it.
The problem is it appears that Kelly Robin is here, but has not been able to chat. And so I don't know whether they gave up because they
I
couldn't.
wonder if
join.
Yeah.
message her.
So I'm not sure it's worth getting him if they're not, you know. I'm just sending a chat. I'm gonna join room 10 for a minute. It is true. It is the team that analyzes DNA from ancient DNA from mummies and things, and they classify variants. So that was very cool. So I checked out Shingan's room and they were on like their third variant. So I think if we give people 3 minutes, well, now in 2 minutes, if we give people the 3-minute warning, that'll be fine.
Okay, so
And then 2 minutes into that, then just hit the close and that gives them a minute.
okay, so I'll do that at 10:20.
Right. So at 10:20, send the message saying this is your 3-minute warning.
Okay,
And then 10:22, just hit close breakout rooms.
sounds good.
Mona, are you trying to say something to us or you just came out of the break?
No, I'm looking for my group. I was locked out.
Oh, you got not. Yeah, but I'm not sure why that's been happening. You can
Yeah.
just join the room you were in. Do you know your room number?
Yeah, number 7.
Yeah, it should just let you join it.
Okay.
If not, Bree can put you back in.
Mona.
Oh, it no longer has
7.
that in.
Okay.
Oh, you're unassigned. Here, I'll assign you. Okay.
That's group 8.
And no. Yeah, you have to join it now. Mona, you should see somewhere to join. It still says not joined.
Yeah, it doesn't actually let us place
It
them.
doesn't let us, when it
Yeah.
says move to, it just moves them to another
Another
room.
room,
It doesn't. Looks like he joined now.
Should I close rooms?
Did you send the warning?
I did.
Okay,
Oh,
yeah, then
okay.
close the rooms. Yeah, sorry. We just got him back in. I'm not sure I assigned rooms to report. I've not been good about that, but I can randomly pick.
Okay.
Okay. Welcome back, everyone. So these were obviously a little bit more complicated. Maybe we can ask, let's see, I think did we ask room 1 the first time? We could ask room 2. So Marina, do you or someone in your group want to say sort of your experience with those variants?
Sure. We had a lot of good questions. I think we really largely got through the first variant, and the question was, if you have a silent variant like that, how do you not miss a variant like that when you're doing kind of your filtration analysis? Because rightfully so, everyone gets a little bit nervous when you just filter out a bunch of silent variants. So we talked about, filtering by ClinVar likely path and path variants, and that this had a lot of ClinVar entries to be able to catch a variant like that, or like really specific phenotypes, like progeria and lamin A. We also had the question of when you should use PVS1, for example, versus computational prediction codes for what type of splicing variants. So we were able to discuss that. So kind of lots of good discussion on this interesting one that you picked, Sharon.
And I should say that the allele registry is starting to link PubMed IDs. Something else I've noticed is if a variant has a lot of PubMed IDs and it's silent, there's probably a reason it's worth looking at a few of those papers. I did think this was a good example because this is one of the classic progeria. I mean, it's an incredibly rare syndrome, but this is one of the classic variants, yet the predictors don't do, but it's, silent. The predictors actually don't do so well. Room 9, any comments about this one or any of the other ones you looked at?
Yes, I think people asking if we can use other tools, not only like splicing AI or the RAR-seq, but I think unfortunately, yes, but I think the first I don't remember the name, but there is a, let me see, like a mask and scan, or I think they are not free anymore. So but if people can access, that would be great.
So Max and Scan was one of the first. It is quite complicated to actually use and to sort of understand the output. So I would say from the ClinGen perspective, what we did with the computational predictors, because a lot of people think ClinGen says use Rebel, and we didn't try to pick one. The paper that came out for the missense predictors said, this is how we analyze the variance. This is sort of the statistical test we put them through. Here are all the variants we tested them on. If you have a predictor that works just as well, great. But here's sort of how to test the predictor. Of the ones we tested, here are the ones that had, a relative strength to strong versus moderate. That work is sort of going on behind the scenes with splicing. I'm not in the splicing subcommittee, but I think I've seen an early draft of the paper. So we do hope, and again, I don't think we're going to pick one tool. The other reason we have to be careful about picking tools is sometimes they become commercial or, maybe more expensive to access. But what we try to generally do is say, what's the best way to judge a tool? I think generally in ClinGen, if there's experimental data, that trumps the computational predictor. So for the splicing, obviously, this is a well-known variant. People had RNA that could show it misspliced. But, I think that, that's, I'm not an expert on splicing, although my lab now, through coincidence, is now studying splicing. And what we're discovering is there are a lot of variants in introns and even silent variants that are missed by clinical labs that Splice AI or something predicts may in fact be splicing. And we're now testing those in the lab for one of the cancer genes. Any other general questions? We're actually doing great on time, which I'm delighted to see. Thank you to all the group leads for that. The last one, and then we don't have anything long at the end because I know people, it's evening now in Africa. So we'll just sort of wrap up and say thanks to folks again and just remind people about how do you sign up for an affiliation and things like that. And Deb, maybe if you send me that link that you or Matt created, we can also put that in the chat at the end. The last one, of course, are missense variants. We give you a lot of data just again, so you can try the missense related codes. So here are the computational predictors play a role. Here, people often do functional assays on the common genes. And we've provided the people in your rooms some of that data, so you don't have to go find it. But it gives you sort of a different set of codes that you don't necessarily use if you don't have a missense variant. Again, if any of you get bumped out, we have found that if you leave the meeting, let's say you have to go leave the meeting and come back in, Fareed can easily reassign you. Just come back to the main room and we'll reassign you. But again, you can join the breakout rooms. And I think anything else, Deb, any of the other leads, any other major issues come up?
No, I mean, we got through the first one and part of the second one. So, I mean, I do think the issue with the two splicing programs not confirming the functional took quite a lot of time, but it's a very real situation. So, yeah.
Yeah. Okay, then we will put you into your rooms for the last time.
Okay, here we go.
Hey, Matt.
Hi, Sharon. I was about to join Deb's room. I was in Marina's room last time. I'd say it worked quite well. I mean, it is a little bit, it does feel a little bit strange because sometimes you have to accept there's going to be a little bit of white noise because, like silence, because when people are in, particularly like when the people are all in the room together, they're talking with each other at their end, but we're not hearing them necessarily. And then they come, but then when people started looking at the variant, they did all come on then and start asking questions. So it's really productive.
Well, there is one room where they're actually in a room.
Yeah.
Right. Yeah. So there is a room where there's a whole bunch of people in a room. So most of them are not. Yeah. Most of them are on individually, so you hear them, but I forgot that. Amanda said, hey, we're all in a room. I said, that's fine, as long as we know which, as long as you're all on the same team, you can be in the room. Yeah, I forgot that the first one, when I picked that one, you know, I didn't realize, like, oh yeah. Splice AI really, this is not a good example for Splice AI, actually, for neither of them, but I should have picked, because I'm actually sort of a believer in Splice AI. I probably now caused the whole continent of Africa to not. And I was telling Fareed that, Mona, do you need to be put back in a room? Because Mona had problems before.
I can't add them back into the room. They have to add themselves.
Okay. I meant assigned. Yeah. I didn't mean is yeah, no, that's what everybody's assigned now. So that's not a problem. Yeah. And so the other thing is that room 10 is what I thought it is. People classifying variants from mummies. So
that's fascinating.
That's totally cool. It's the ancient DNA group from the National Museum of Egypt.
Oh, that's amazing.
Yeah. So I went in and just was like a fan. I was just like, is that what you guys are doing? So I used to always study Egyptology when I was a kid. Okay. Salia, are you here? Do you need any trouble? Do you need any help getting into a room? Some people just, you know, if they walk off during the break, then they'll know to go in when they get back. Okay, well, okay. So the reason I wanted to call you out is, do we have a slide or something, or can you pull up your slide? I think for like the very end, just sort of a reminder that number one, you know, Mark and I talked about maybe going to June 1st, because not all these people have access to internet all the time or whatever, but that we really want to encourage them. So do we have like a slide that just says, you had that slide that had the login for the test. And so does the test and the real one require two separate logins?
Yeah, they do. Yeah.
Okay. So maybe just review that. And, you know, if they really want another test, it'll be sort of like this one. But if they want to work in the production, they need to request the login. And then the thing about creating an affiliation. I think that's probably the best thing to sort of end with.
Yeah.
So I think, Fareed, for you, I would probably at 50 minutes after the hour, give a 5-minute warning. Because we really only need like 5 minutes at the end.
Okay, I'll do that. At 10:50. Okay.
Yeah. I never believe in sort of long wrap-ups, but I think, Matt, it would help to just sort of end with next steps. And also just remind people that we have lots of educational material on the website and the help document. I was glad you brought that up. I'd forgotten to emphasize that. So.
Yeah, that's one thing we haven't actually done in this session. I realized that we haven't actually shown people the output on the website where they can go and find the curations that have been done. We actually didn't mention that at all. Like,
oh, the curations that have been done.
Yeah, and where they can go and see them.
Well, can you do what? Well, you can either do what Deb did or just have them both open. So maybe if you can show your slide about how to register and then remind them that they can see, you know, they can link to any classified variant from our website, but also that the teaching materials are on the website. And that, but the other point I was just making is that even when you're in the VCI, there's help documents.
Yeah.
So I was just trying to emphasize, you get in and you get lost, you know, you hit the help thing and it, you know. Because I think that we You know, because that's new, that's not in all of our slide decks. The
Yeah,
help.
that's right.
So we sort of have to remember to sort of add a slide about, I mean, you did at the end, but I just meant it's not in all of our slide decks because it didn't used to exist in any robust way. So that would be sort of important for us to just remember what we're doing.
Yeah, it was one of the things that Christine did before she went on maternity, is that it was her, Ingrid and Rachel, they went through and completely rewrote all the help docs. And now they're on top of it. Every time we do a new feature, they go in and amend the help docs straight away as part of the release of each new feature.
Yeah. But yeah, no, I agree. I think that's great, but I think we're not used to talking about it. So, you know, just sort of doing that. And it's great that we have the volunteers. So happy that we were able to do that. Yeah, I think it made a huge difference to have them work in small groups. You know, and some of the groups are quite small. That just has to do with how people signed up. A couple of people, you know, a couple of groups just signed up very large numbers, and we didn't really have a way. We tried to say, you know, how many. Some of the labs really did just sign up 4 people, so they have 4 or 5 people in the room. But I just think having the volunteers was just super important.
I mean, I was in the room with Marina, and of course, Marina was amazing.
Well, we gave her one of the biggest. We gave her one of the biggest rooms for that reason. I was like, I said to Fareed, Deb and Marina, get the biggest. That's why if I was going to do it, I had already picked out the smallest room, but then we got a few more volunteers. And Chang'an very politely went, so Chang'an, thank God, actually went through them all yesterday, like code by code. And he was like, Dr. Plum, you know, these don't agree, you know. So he said, so like late yesterday, you probably weren't on that email. I sent all the coordinators, like, hey, here's some additional points for each room. So fortunately, he went through and discovered some of these surprises. So that they weren't caught up totally off guard, like, oh. Oh, and then Chang'an's also the one that discovered the P10 release version 3 yesterday. And I'm like, is it in the C-spec registry already? It's like, yeah. Like, okay. So then he actually went through and reclassified the variants and said which code would have changed. So I actually think it's a good example of go to the website, go to the registry, don't go to the papers, right? Because that P10 paper was published years ago, probably on version 1. So yeah. Okay, so help documents, website, C-spec, I'm glad May was the variant, and I will answer that. So that's a question, Matt, for you. So you know how Nomad and ClinVar now, both, Nomad has a nicer graphic, but both of them show you the variant in the context of the gene, right? They'll like show you the other nearby variants. We do, I remember us talking about years ago, we do something about that where you can pull in the nearby variants, but do we have, I mean, I think for the new VCI, having some kind of display, you know, and you may be able to just kidnap the Nomad one, you know, you might be able to share the code for these novel variants. So even though their variant may not be in ClinVar, they could still see their variant, you know, surrounded by, let's say, the ClinVar variants.
I think that so ClinVar have got they've got the new lollipop.
Yes, they have their lollipop. I have to say, I like Nomad look a little better, but we could look at either of them.
Yeah.
But I think something like that. See, the problem is with ClinVar, it has the lollipop, but you can't put a new variant into it. So let's say there's a variant in the allele registry that's not in ClinVar, but you want to see it's 3' of these benign variants. Is there a way, that was a question that came up, I think, after the first round. Is there a way to see your variant in its context? And so that may be something that could be really useful.
Yeah, because at the moment you can only do it if you link out. Like we provide link outs that automatically take you to the region in different resources, but we don't do it within the VTI itself.
Ah, but we do take you to the region, even if your variant's not in that resource.
Yeah, so it's just based on the location. So those genome browsers, for instance, UCSC, like it'll just based on the location, it'll automatically just take you to a region that's 30 base pairs up and down from the region. So you'll be able to see.
So that's part of, I think. So Rolanda, who I think is still on by phone, I know, I think she's driving, and Deb, I think, worked to put together some questions for afterwards. So Rolanda, are we going to send that by email?
So the certificate will be linked to the feedback form so that we can ensure that the folks fill in the feedback form.
Okay, awesome. So to get their certificate, they'll also get the feedback.
Yeah, but we have some time, we can do that. We can finalize that even by next week. So.
Okay, awesome.
Sure.
Because I know Deb also had some questions, but you had really good ones. So let's see if she had any. Okay, so Freed, I may change my mind and give us a few more minutes. Have you sent them any warning yet?
No, I will be. Did you said at 10:50, right? Did you want me to send it earlier?
Yeah, we probably do need 10 minutes at the end. Do you want to give people a 4-minute warning now?
Sure.
What happened? Oh, we're viewing Matt's screen. Okay, sorry. I don't know if you meant to do that, Matt.
I did it, but it didn't show the one I wanted it. So
it just showed a desktop.
That should be showing. Yes, I wanted to show you this. I pulled out those slides and put them at the end. So we just work out what order we want to put these in. So we've got this one here, which is just the help documents and the contact the VCI. We've got the reminder about the affiliations. we've got the individual registration, how they register afterwards. And then I've also just put a slide in, we just have the website, just to remind them they can go and get information for like training materials and everything else.
Okay, yeah, I think that order is probably fine. So I'm just going to say a couple things first about the certificates and the feedback form, which will come next week via email. And I think the main thing when you show that help thing, because the first thing you see is the help desk, which is different, right? So I think just sort of maybe circling that these other things are going to give you help on the VCI, not help connecting, you know what I mean? Because you sort of see that. And most people were able to stay all the way through, which is good. It was late there. Okay, I would hit the close button.
Okay.
Oh, gosh, this is annoying.
Hey, Amanda, you're back.
Can we just ask if we are going to get links as usual, I'm assuming, to all the documents?
Yeah, we're going to review that. So welcome back. I am delighted that almost everyone could stay for the whole 3 hours. A 1 minute huge thank you again, round of applause, especially to the team that put this all together, Fareed, who was Zoom master throughout all this, Mark and everyone. And again, a huge thanks to the volunteers in each of the rooms. Many of them didn't know I was going to make the variants as complicated as I did. The main thing we wanted to spend a little time on is Matt's going to just re-review a couple of slides about now that you've tried it, sort of how do you get on? So Matt, turn it over to you.
Yeah, so I wanted to so this is really the next steps for all of you. So one of them is if you want to find out any more help, like as I said earlier, if you go to the header, and that's in either the curation or the test site, or even actually in the instances that you've been using today, if you go and click on the website and do the pull down, you'll see here that you can see help VCI GCI. So if you click on that, there's really is, now I said this earlier, but there is really extensive documentation here that we keep up to date with all the latest features of the VCI. So if you go there and have a look, it really does walk you through exactly how to use the VCI. It's all in Gitbook. It's like really clearly laid out for you. And then also the SOP for the VCI, if you click on that, that will take you to the website and it will show you the advice from the variant curation team on how to do variant curation according to the SOP. And then, of course, we also have the help desk email that takes you through to us at Stanford, the team here. If you've got any questions about how to use the VCI, we're here to support you on that. A reminder about affiliations. So it's similar to the way that you've been using it today. If you have an affiliation within the interface, you can all work together and see all the curation together. You can classify the variants together and see each other's work and work together and curate as a team. So we'd really encourage you to sign up as your lab or group or institution. I know I've got a bit of an echo there. Also, yeah. Or institution, and all we need from you really to set one up is the name of your affiliation, and that's just the name that's going to then appear for that affiliation within the, your group within the interfaces, and a contact person, and it's that contact person that will be in contact with about who, for instance, should be added to that group, because you don't need to make your mind up on that straight away. We can set it up for you without even knowing who the members are. You can add those in later. And there's the contact sheet, which is, I was going to post it into the chat actually before I started this. I don't think I did. So someone else can post that.
I think Deb did.
Oh, thanks, Deb. So you also, although you've registered, we pre-registered today for the interfaces for this, in order to use the production and test sites, you will have to do that registration again for the production and test, but welcome you to do that anytime. All you need to do again, you go to the login in the header. And when you do that, what you see on the right-hand side here will pop up and you'll be able to see and you click on create account and then just follow the instructions. And then you'll be registered to use the VCI, both the test and the production site. I should say the test site gets wiped periodically, so you don't need to worry so much what you put in there. The sites that you use today, I think we said, Sharon, they're going to be up for 6 weeks or something. Yeah.
So the ones today are really, really temporary. If you want to play with the test in the future, you should use the real test. But we'll keep these open for 6 weeks in case someone wants to go back and show someone else on their team what they did. But these were just meant to be temporary for the workshop, and we've reused them. For example, we gave this workshop in India.
And then the final slide I have is just want to remind you that you can see the entirety of the output of all the variant classification work that's been done by ClinGen on the website. And then browse what other folks have done and see those comprehensive final classifications. But there's also an awful lot of training materials on the website. We have a huge volunteer workforce as well, and so we maintain lots of training materials for them, but also for the VCEPs. And so if you go to the website, you can see all the information that we have on all those different training materials. We actually also have quarterly training sessions on how to use the VCI, and I think one's coming up soon in the next few weeks. Is that right, Marina? Do you know, is that the beginning of April sometime? But people could also sign up for that, and during that session it's kind of like a longer training session. I think it's like an hour or something where you go through the VCI in more detail. Is that right, Marina? Is it I think it is coming up in the next few weeks. Yeah.
We haven't set the training session yet, but I just gave some availability dates for myself. So it should be in April sometime. And if you're in my group, it's basically the same thing where I walk you through live how to use the variant curation interface.
So I did want to answer an important question in the chat, which is an affiliation an expert panel? So no, if you want to create a ClinGen variant curation expert panel, please go to our website. There's a whole set of instructions. It's a 4-step process where you will create an application. You'll tell us what the disease entity. We do require those to be multi-institutional. And so TP53, myeloid malignancy, hearing loss, some of these cover a number of genes. Some of them cover single genes that are heavily tested for. The affiliations you're requesting are if your lab wants to work together on the VCI. If you become an expert panel, we create a ClinGen variant curation expert panel ID in the VCI. So this affiliation is not creating an expert panel. It's saying, hey, my lab wants to use the VCI. We want to work together. So like some of the research labs in the United States use it. If they have variants they want to classify, they work together. They then submit them to ClinVar under the name of that research lab. That's if you want an affiliation in that way. If you want to form an expert panel, then I would look on our website and reach out to the oversight committee to get advice about that. Okay, the last really important thing I wanted to say, and then Deb, we'll see if we have time for your question, is that Rolanda and I and the organizers are giving out certificates for completing the workshop, and delighted how many of you stayed through the whole thing. We are going to send a survey, a feedback form with that. This is our first time trying to do it this way. We did it in person, as I said, in India when I was at a medical genetics conference, but this is our first time sort of doing it this way. So we really want some technical feedback, but more importantly, we want feedback on what you learned and how we could do this better. So those emails will probably go out next week, and your certificate for completing the workshop will be attached with the feedback form, just as H3Africa has done for other events. Any last minute sort of critical questions? We're right on time, which I'm just thrilled. I'm not sure I understand the question, but basically a well-established functional assay, it could be that less than 50% is a well-established assay. So I'm not sure I understand the question, but that is one of the things ClinGen gives a lot of guidance on in terms of what functional assays to use. That information, again, that web page. So again, just answer that early question. I'm classifying a variant, a gene for which there's not an expert panel. If you go to the sequence variant interpretation page, we give you guidance for those codes. If there is an expert panel, you go to the criteria specification registry. And in fact, many of those have also published papers. The most updated information in ClinGen is always on the website. So P10 is a great example. They updated their specifications on Tuesday, and the website, the CSpec registry, had the new specifications. The paper they published was published, I think, 3 years ago, right? So if you just go to the PubMed ID, you're not getting the most up-to-date information. On the other hand, that publication may describe in more detail how did they look at functional assays and how did they make the decision they did. So with that, we're on time. Thank you all again for participating. We're delighted it all sort of went off without any major technical hassles. And thanks to the large team that did that. And please look out for your emails next week about the certificate and the feedback form. And good night to all of you in Africa, and good morning to all my colleagues here in the US.
Thanks very much.
Thank you guys. Bye.
Bye-bye.